Good afternoon, everybody. Thank you for coming. Let me take you through Kiji Therapeutics project. So, we're addressing autoimmune diseases with a proprietary approach using multi-gene engineered cells, in this case MSCs. We aim to do a proof of concept clinical study in aGvHD, and I'll come back to that. And we have plans to extend that also to a couple of other indications, IBD and skin indications. And we're looking for an extension of our initial seed into a Series A that would enable us to deliver the milestones that you'll see down the road. I think we bring a technology that is anchored on a couple of well-established things, and I'll come back to that as well, which is the safe and utility and efficacy of MSCs powered by gene transducing them to optimize the therapeutic effect. We have a strong team with experience in cell and gene therapy and a strong advisory board. An advisory board that gives us the advice on cell therapy, on gene engineering, but also on the indications, i.e. PhD and IBD, that we aim to deliver our products in. Now, let me take one step back. Why do I believe so passionately that we are doing the right thing, that we are giving a great opportunity to the cell and gene therapy space and bringing a product that will bring significant value to patients? MSCs have been around for some time, and they have a clear safety profile and a clear value. And they've now been approved, in addition to having been approved in Japan and elsewhere, in New Zealand, in Canada, extensively used in Europe. They've now been approved by the FDA for aGvHD. Mesoblast has achieved that approval, and it's been an anchor point in the cell and gene therapy field. I've written an editorial about that as well. Now, we also have other companies developing MSC products, and I would highlight Cynata, who's actually developing a iPSC derived MSC product also for aGvHD. So taking the use of MSCs to the next level of the CMC approach, I would say, with an improved manufacturing process. And then what we aim is to kind of layer on that what it really is missing to the MSCs, which is the power of a well-targeted and potentiated mechanism of action that will deliver a therapeutic effect. And so you can see here the comparison between the now approved product, the in development product, and our proposal of the engineering approach in the end. And so this gives you probably what I would call the three generations. The first generation, which is wild-type MSCs, and Ryoncil is clearly the example of that, and it's paving the way in the market for us. And then the second generation, be it a improvement on the CMC like Cynata is doing, or, as we aim to do, a biology improvement by providing a stronger therapeutic benefit through the engineering. But really the vision, and this is what we aim to do, is to bring that together into an iPSC source, MSC cell that we're able to engineer for optimal therapeutic effect. That will give us the optimal biology, obviously based on the right quality, but with a capacity to produce an attractive cost of goods and large capacity, which will be the position that will take us to target large indications like IBD. So, but what is the key value proposition that we bring is really engineering the cells with two things, CXCR4 on the surface and IL-10 being secreted. That is done through a lenti transduction with the bicistronic gene, which is a proprietary approach, and really is engaging the cells in two things, making them to go where they should go, and making them deliver what is needed to provide an immunomodulatory stimulus by the IL-10 secretion. We have a manufacturing process that you wouldn't be surprised, it's fairly standard. Currently, we have an adipose-derived source, which select, expand, transduce, and formulate off-the-shelf cryopreserved. But that's our GMP existing process, but we also have an R&D process using an iPSC source that we differentiate, that we transduce and characterize. And we do have R&D grade of the same quality, of the same characteristics, of the same functionality that our GMP grade adipose-derived. So that will enable us, with the adipose-derived, to go into GvHD, and we should be in GvHD by the end of this year, beginning of next year. We're now actually building the documentation to submit the IND or the clinical trial application, to be more correct, since we'll be doing this in Spain. But we also are doing the research for the preclinical package for IBD and psoriasis, which we'll aim to do with an iPSC-derived MSC source, and we have recently established a collaboration with a pharma company that will do R&D with an option to take the product for central nervous system indications. The study that we aim to do in GvHD, which will be a proof of concept that we do deliver an optimized therapeutic effect with cells that already are recognized in that indication, will have the standard 4 administrations weekly, and the normal dose of about 2 × 10⁶ per kilogram in multiple formulations, enabling to be immediately administered IV upon thawing without having the complexity of cell preparation. And we will be doing the assessment that is standard in GvHD in terms of overall and different responses. But we will do 2 things which are very important, is not only the short-term assessment, but also the long-term assessment, because I think one of the key elements that we have already seen with wild-type MSCs, and we're seeing it stronger in our animal data, is the long-term benefit, the sustained benefit, enabling less administrations, even though we are also doing gene editing for increased tolerability, allowing multiple administrations in case we need. The markets we're addressing are significant ones. We don't need to go on to that. I would say that we have the opportunity to go for a smaller market with an adipose-derived donor-based approach, but targeting a very large market, IBD and psoriasis, with an iPSC source, really enabling us to get a value proposition from cost goods to clinical facility that is attractive and delivers therapeutic benefit. But let me also give you a little perspective of the reason to believe. Why do we are so strongly confident that we will deliver value on top of what MSCs have already done, is that we have multiple animal data showing an incremental in the benefit. And you see here in GvHD, where you see untreated animals, you see the middle curve where you see the animals treated with wild-type MSCs. So you do see the benefit that led the current clinical use and clinical approval, but you see then an increased benefit, a statistical significant increased benefit achieved by transducing those cells with CXCR4 and IL-10. We see the same thing in an IBD, where the standard DSS model that you see here, with the insult, treatment, and you see the short-term effect that we increase the benefit using the engineered cells, but even more interestingly, we see the sustainability of that effect upon repeated administration. You know that IBD is a relapsing and remitting disease, so we model that by re-insulting the animal, recreating a disease, but we did that without retreating the animals, and we saw obviously the disease reappearing. We saw some benefits remaining on those animals treated with wild-type MSCs, but we saw the same benefit, a sustained benefit on those animals that were treated with engineered cells. And we believe that is actually due to an impact on the innate immune system, and we documented that by the profiling of the macrophages and myeloid-derived suppressor cells, really enabling a possibility of a long-term benefit. We also have similar data in terms of biodistribution and increased benefit in a skin model, enabling us to pursue the psoriasis indication. So just to wrap up, we have preclinical evidence in vitro of the benefit. We have multiple animals models showing the incremental benefit with the engineering, and we actually detail the mechanism of action in terms of biodistribution, IL-10 production, and the cell populations, Treg, Breg, and macrophages changed, really giving us a description of the mechanism of action. And this is important because we're not letting the cells do what they want. We're telling the cells to do what they need to do by engineering them and targeting them. So our strategy is to really focus on short-term on the aGvHD approach, which is a kind of a small patient numbers, high willingness to pay, and we have the door open by the bold approach of Mesoblast, giving us an opportunity to really do the proof of concept and have an asset that we would be willing to co-develop, enabling us to start development of IBD, where we will aim to target patients with a macrophage-driven phenotype, being able to do a targeted approach to the clinical development, as well as aiming at psoriasis and the CNS. We're already, as I said, doing that in collaboration. The milestones that we're aiming to achieve with this work is the clinical data in aGvHD. It's developing our iMSC platform to GMP level. We have it on R&D level already, and go into the clinic with IBD, psoriasis, and CNS over the period of 25 to 28, which is really, in a way, a slide of the use of the proceeds. So we see ourselves as a state-of-the-art using the MSCs, but the MSCs, the way they should be used today with the tools that we have to engineer them for optimal effect. And we see the opportunity to go to very attractive, large indications like IBD and psoriasis, and we're obviously, like everybody else, we're aiming to do that with the support of investment, dilutive, non-dilutive, and BT and partnerships. I show you a photograph of an island in between France and Spain, which is Pheasant Island, which is an island that has French sovereignty for 6 months and Spanish sovereignty for 6 months. We wanted to use a symbol of something between France, where the company is established, and Spain, where everything happens for us in terms of R&D and manufacturing. And this island really symbolizes that in between the two countries. Pheasant Biotech wouldn't be a good name, but Kiji is pheasant in Japanese, and is also phoenix, which I think is symbolic for the chance that we're giving to MSCs to really play a significant therapeutic role. So hence the name of Kiji, hence the picture, and hence our project. So thank you very much. Any questions if you want at the end, but I hope you have the same passion that I have for this project. Thank you.